Hello, my name is Robert. I'm a grad student in the Ayres lab where we study infections. And typically when you think of your body dealing with an infection, you think of it using your immune system to kill the pathogen. But we study this thing called cooperative defenses, where you can actually promote health without killing the pathogen.
Hi, I'm Sam. I'm a graduate student in the Ayres lab, and I study how fat can help with a parasitic infection and maintaining health during an infection. And so today I wanted to show you a technique we use in the lab quite often called qPCR. So this is actually a technique that's used to measure the amount of genomic material, so RNA or DNA present in a sample. You may have actually heard about this in the news lately because this is the same technique that doctors can use to determine if somebody is infected with SARS-CoV-2. So the idea just being if you have genomic material such as RNA from the virus present, then you actually have the virus. So I'll show you how we do this in the lab. Adding DNA from samples that I extracted previously, so in this case my parasites, to determine how much DNA is present in each sample in different conditions. I'm adding Cyber to the plate, and Cyber is a reagent that we use that has a dye in it that actually fluoresces with the DNA, so that helps us measure the amount of DNA there. So now I'm putting the plate into the actual qPCR machine, which is the large white block off to the left. So this machine just cycles through different temperatures and actually allows for the qPCR to amplify the genomic material. And so there's actually a computer on the other side that controls this machine, and I input all of the temperature changes that I want it to go through. So these are some of the results that you'll get from qPCR. So this is looking at a very specific gene that we just put into the machine, and these lines here that are going up kind of exponentially and then leveling off are different samples. And so the idea behind this qPCR is you have these cycles, and every cycle you're doubling the amount of genomic DNA present until it reaches this threshold where we can detect it. And so any sample that crosses this line, we then say it's detected at this point. And so the reason this can then quantify the amount of genomic material is that every time you go through a cycle, you're doubling the material. So I have some samples that I put in that are my standard. So I know how many, in my case, parasites, but in the case of COVID, you know how many viruses there were that you extracted DNA from or RNA from and put it in. And then if one of your samples came up one cycle later, you know that it started with half as much genomic DNA or half as much virus because it had to go through one full more cycle to reach this threshold. And so these are the results that you get from the qPCR that I ran, and then this is exactly what you would expect in a clinical setting if you're testing for COVID.
In order to study bacteria, a standard technique we use in the lab is to grow bacteria on nutrient agar plates.
Hey, my name is Grishka. I'm a microbiologist in the lab, meaning that I study bacteria and viruses. My research focuses on how gut pathogens cause disease and how these pathogens evolve over time during infection.
Here, I'm demonstrating how we grow bacteria for experiments. Using a sterile toothpick, I can transfer bacteria from an old plate to a fresh agar plate. I streak several times in order to isolate individual colonies of bacteria. Next, we put these plates into 37-degree incubators. E. coli grows very fast, dividing every 20 to 30 minutes, meaning that overnight we'll get a full plate of bacteria. These particular bacteria that I grew up express a red pigment, meaning that when we expose these bacteria to UV light, they actually fluoresce this red color. These bacteria are very useful for our experiments because we can use this red fluorescent protein to visualize bacteria during infection.
7, 6, 5, 4, 3, 2, 1.